Hello, my name is Christopher Henriksen, and today we'll be talking about tRNA, the adapter hypothesis, and Francis Crick. So how does genetic information flow from DNA to protein? We know proteins are made up of amino acids, and amino acids are characterized by their side chains, whereas nucleic acids are composed of a string of nucleotides whose sequence allows for hydrogen bonding between the bases. This makes nucleic acids good at recognizing each other, but poor at recognizing the varied side chains of amino acids. So this apparent lack of specific recognition of amino acid side chains is what led Crick to conclude that amino acids would first become attached to a small nucleic acid, a tRNA, the adapter molecule, and by base pairing with the template would carry the amino acids to be lined up with the template. Furthermore, transfer RNA serves as a link between the genetic code of mRNA and the amino acids that make up a protein. Each tRNA attaches to a particular amino acid and carries it to the ribosome, where tRNA adds its amino acid to the growing polypeptide chain at the position specified by mRNA. Each tRNA is capable of attaching to only one type of amino acid. Because there are 20 different amino acids found in our proteins, there must be at least a minimum of 20 different types of tRNA. In fact, most organisms possess at least 30 to 40 different types of tRNA, each encoded by a different gene. To further make sense of what I just said, we have our mRNA that has been synthesized from DNA, left the nucleus, and attached to the ribosome. Our codon of interest is GAG. Then our tRNA comes in, boop, and it pairs with GAG to CUC, anticodon. And this will encode for a protein or amino acid, an amino acid to be added to the growing polypeptide chain, which we can see right here. And as more tRNA molecules come in, the longer the growing polypeptide chain will be. Now, there are a couple of things that could allow for tRNA to not be able to complete its task. There's an enzyme called aminoacyl tRNA synthetase, which correctly pairs the tRNA with the amino acid that it needs to bring. Now, if there was a mutation between this enzyme subunit that did not allow for the right or any amino acid to pair with the tRNA, then it would not be able to bring the amino acid to the mRNA, and the polypeptide chain would not be synthesized.